Good afternoon, everyone. I'm Jennifer Warner. I'm a new technical program director at Current. I'm just about 3 weeks into the job, so be patient with me. I'm so pleased to welcome you all to this month's Third Coast Water Seminar. I want to take a moment to acknowledge the amazing team that we have behind this really powerful event series, including Northwestern University, the University of Illinois at Urbana-Champaign, Argonne National Lab, the University of Chicago, and University of Illinois at Chicago. This team really is leading the charge on water innovation for our region and brought us an awesome speaker today, Dr. Rachel Poretsky, who will highlight her work on the regional efforts for wastewater covalence for COVID-19. So there's a few housekeeping things. For those of you who have participated in this event series in the past, this might be familiar. For those who are new, please know that there are two tabs in the panel for the Zoom platform to offer questions and comments. We prefer that you provide your questions in the Q&A box and your comments in the chat tab. We will be taking a look and keeping a close eye on both. The event is being recorded, and the recording link will be shared on Current's website and YouTube channel and will be sent to all of the attendees here today after the event. We have a very simple agenda that will highlight a significant speaker from the water research community, Dr. Rachel Poretsky, and gives your audience a chance to engage through questions. I'll start with a brief intro today about Current and what we do, keep it really brief. Then we'll pass it on to one of my Third Coast colleagues, George Wells, to introduce today's speaker. She'll present for about 35 to 40 minutes, and we'll have about 10 minutes for Q&A at the end. So be sure to send those questions in the Q&A box. And we'll end with some closing remarks, and there will be an evaluation little survey at the end too. So please make sure you stay for that. We'd love to get your feedback. So a bit of background on Current. Is this good? Yeah, here we go. Current's Chicago's water innovation hub. We've been around for just over 5 years, so still fresh and new. We're tasked with the mission of growing an inclusive blue economy, accelerating innovation, and solving persistent water challenges. You know, why are we here? For the most part, hold on a sec. We're here because we believe that we need access to healthy water to drink, and we want less flood water in the basements. We want healthy people, we want equity, we want healthy environments, both locally here in Chicago and globally as we deal with the effects of climate change. But we also want to address the important economic disparities by leveraging the innovation happening in Chicago to get Chicagoans ready for blue jobs at homegrown companies solving global water challenges. The challenge that we're seeing here in Illinois is that people are not rallying fast enough around innovative ideas that will help us protect our health and environment. This is why Current builds partnerships and collaborations that allow us to solve the problems across sectors and take some of the risk out of the new and innovative approaches. One way that Current does this is by being an unbiased advocate for the best water solutions and policies. We're always scanning for new innovations in water and assessing new technologies, often making recommendations to our partners or running the pilots ourselves. More than just a connector, Current often tests new technologies in-house and with our partners. On the right, you'll see one of our team members pulling water quality sample from the river as part of our H2 Now Chicago real-time water monitoring pilot. This pilot launched to the public in the fall of last year, and we're gearing up for another season starting this spring. In fact, just yesterday, we had our staff and our partners at MWRD out on the river installing probes. So we're busy testing and calibrating now, and we hope to launch season 2 here this summer. We're also very excited to continue working on some emerging areas of interest, including our sewage surveillance work. We'll hear a little bit more about that today, looking at strategies to reduce nutrient contamination in waterways here in Illinois, capturing and using renewable energy from wastewater heat recovery, and eliminating PFAS and other forever chemicals that threaten our water quality. And finally, tying much of this work together is a regional strategy to grow an inclusive and equitable blue economy, where anyone can develop an interest in water and follow that interest into education and high quality jobs. This strategy, which is being developed by Current and our partners, also uplifts and promotes water stewardship from our corporate community here in Illinois, ensuring that we are striking a balance between those leveraging our water resources for economic growth and protecting those resources for the future. Now, I'll wrap up that brief intro and hand it over to my Third Coast collaborator, George Wells. George is the Associate Professor of Civil and Environmental Engineering at Northwestern, and George will introduce us to today's speaker. George?
Thanks very much, Jen. Really a pleasure to introduce our speaker for today. Dr. Rachel Poretsky is an associate professor in biological sciences at the University of Illinois at Chicago. She's been at UIC since 2013. Prior to joining UIC, Rachel obtained a BS in biology from Brandeis University, then did her PhD at the University of Georgia with Marianne Moran, got her PhD in 2008, and then was a postdoc at Caltech in the Department of Geology and Planetary Sciences with Victoria Orphan, and then at Georgia Tech in environmental engineering with Kostas Konstantinidis. Rachel has done a variety of really impactful and really exciting work on environmental microbiology, specifically focused, at least in large part, on aquatic systems, on lakes, the open ocean, rivers, on wastewater treatment plants, and the like. And she's really worked to pioneer or really develop this nascent field of wastewater-based epidemiology. This is a really exciting and rapidly developing area and will be the focus of her talk today. So without further ado, Rachel, thank you so much for joining us today, and I'll turn the floor over to you.
Great, thank you. Let me get my presentation up. Okay, thanks, and thanks for having me here today. I have on my first slide my email and Twitter handle. Either are open and available for anybody to reach out to me if you have any questions or ideas or concerns or anything to follow up with me. I am happy to talk further. So I actually took the SARS-CoV-2 part out of my title here because this wastewater epidemiology project continues to grow and expand. And initially we had tunnel vision like everybody for COVID, but we're now thinking of it in terms of an established program that is useful for all sorts of public health concerns and ecological concerns as well. So I'll sort of end my presentation with that, but I'll start with a background on wastewater-based epidemiology to begin with. And I have here some general information about what it is. I think a lot of people are now more and more familiar with this idea, but for those of you who aren't, the premise is that we can use wastewater as an epidemiological tool by monitoring what is in the wastewater, and it is a composite biological sample, the ultimate composite sample, because everybody contributes, and this has actually turned out to be a really important and useful tool for COVID, because the virus that causes COVID is turns out to be shed in feces, but also in mucus. If you blow your nose in the shower, it all goes into the wastewater system, and people seem to shed this virus, whether they have symptoms or not. And I'm sure we can all think back to the beginning of the pandemic when first when tests were not even invented yet or available yet, and then when they were available, but being able to get the results was a challenge. Sometimes it took days to get PCR, to get an appointment to get a PCR test, to get the PCR results back. We can even think back to recent, the Omicron, the first Omicron wave this winter, when test availability was again, there was a shortage of testing. And so Wastewater doesn't care about any of this. People continue to use the toilet and contribute to wastewater samples, regardless of whether they've been tested, whether they have symptoms. And initially, when people first started developing these tools for wastewater-based epidemiology for SARS-CoV-2, there was evidence that that wastewater could be a leading indicator. And now we know it varies. It varies from sample to sample or from site to site, depending on all sorts of things, the viral dynamics and which variant is the dominant variant that week. There's lots of confounding factors that go into this, but in general, we found that there are a good number of times when we can see trends in wastewater prior to when we see the same trends in clinical data. So I put at the very bottom some facts. This actually comes from a commercial company that we happen to get a lot of kits from, but they have a couple of facts about how it can be a leading indicator, how it's a very sensitive method. So theoretically, 100 people, we can detect 100 people among 2 million, 100 people with infected people within a population of about 2 million. And we can get good epidemiological trends, and maybe I don't know if it's most importantly, but importantly, it's cost effective. So if you factor in the population, we've calculated that it can be cents per person versus dollars per person for clinical tests. Right now, there's a lot of places that are engaged in wastewater surveillance for SARS-CoV-2 across the world. A colleague at the University of California in Merced maintains this database, the COVID-19 database, that has a running tally of current efforts in wastewater based epidemiology, and many come from universities, many have public facing dashboards, a lot of countries are represented, and a lot of the world's population, a fraction, but a good fraction of the world's population is being monitored through wastewater surveillance. And I have to say that this has been probably one of the most collaborative projects I've been involved in my research career, because as this is being developed here in Chicago and here in the state of Illinois, it was being developed all over the world at the same time. And we really built this community where people were leaning on each other and sharing method development and troubleshooting. And as it was happening, you know, in a lot of projects, we generate data, we sit on it, we ruminate on it, analyze it, publish it, and then talk about it. But in this, we're all working together across the world, and it's been really an exciting effort. So the scheme is that we collect samples from either water reclamation facilities or sewer systems, congregate living facilities. We have a variety of sites that we work with. We transport them cold to the lab. In my lab, we do the PCR-based analysis. We also do sequencing at Argonne National Lab. I'll talk about that a little bit too. And we analyze the data with public health data. We model it, we integrate it with other public health data. We communicate with our local public health agencies. We report nationally, we report to the National Wastewater Surveillance System, which is run by the CDC, and our local partners, the city and state departments of public health, as well as local Department of Public Health departments of public health across the state. We're also working with data and modeling team members who help analyze the data and incorporate various modeling aspects. So you can see from the logos that a lot of the partners are the same as the Third Coast Water partners. There's a lot of overlap. our team is large and amazing, and this is just a fraction of the folks who are involved. But I put some of our leaders on this slide. So Sarah Owens is at Argonne National Lab, and she runs the sequencing part of this work. Charlie Khaled is the senior research scientist. Aaron Packman, many of you probably know him from Northwestern University, kind of leads our analysis and modeling component of the project. Melissa Pierce is relatively new to our team. She was a postdoc in my lab for a number of years before turning to the field of data science. And so we, in the year since she left my lab and worked in the realm of data science, we brought her back into the fold to work on the data science portion of this project. And Laura Clements is our project manager. But there are a lot of people. We have, I sort of highlighted in bold here that overarching teams that are crucial to the success of this program. And so we have the folks who are involved in sampling strategy, design, implementation, the folks who are involved in the detection methods, the molecular methods, the microbial ecology. George Wells is actually one of those people. The sequencing analysis. We have folks who are involved in the social vulnerability assessment, data analysis. I mentioned public health planning and response. And I'm going to give you a little bit of an overview of how this project started and where we are now. So, but briefly, we began in October 2020, so pretty early in the pandemic, and expanded statewide in 2021. So our team really got together because we had this diverse group. We all kind of knew each other. Well, not everybody, but a lot of us knew each other, and some of us had worked together previously in microbial ecology, wastewater monitoring, urban aquatic systems, modeling, data science, and many of us had worked in the Chicago area before at the scale of neighborhoods in the Chicago River, Lake Michigan, and in wastewater. And so we brought together our expertise in water and wastewater and in modeling and data science, and built this program that really involves lots of aspects of this work. So this work all began in Chicago initially. So in August 2020, we received a science grant from, we became one of the first science teams, a seed grant from the Discovery Partners Institute, and to actually look at urban streams and urban aquatic systems for signals of SARS-CoV-2. But quickly after that grant was funded, we got a big award from the Walder Foundation as part of their CAN initiative, their Chicago Coronavirus Action Network, I think is what CAN was stood for. And so our project was called PCAN. It was a prototyping wastewater epidemiology program in the city of Chicago, where we started with, in collaboration with the Metropolitan Water Reclamation District, with the water reclamation facilities in the city of Chicago, as well as some local sites, including Cook County Jail and O'Hare Airport. We slowly brought other sites along in the city, but the idea was, could we make this work in a large urban environment, especially a place where we have a few water reclamation facilities that serve millions of people. So it's really an issue of scale where we're trying to identify trends in this disease burden in this in the city by essentially surveilling thousands of people at a time. And so this prototyping project took off was funded in September. We got our first samples in October, and all this work was initially done in Dr. Abhilasha Srisa's lab in the School of Public Health here at UIC. She was already set up. She has a contract with the city of Chicago to monitor the beaches. So every summer you probably see the daily beach reports should the beach be closed? Is there E. coli and enterics in the beach water? So her lab does that summer beach monitoring, and so she was really well set up to take this on. And it was the off season for the beach, so all this work began in her lab. In May 2021, so about a year ago, we expanded to the entire state of Illinois, and the program has been growing since. So we're currently in over 65 sites in about 49 different counties across the state. And so this is not super up to date, but generally up to date, shows some of the participating counties of our Illinois, we call it our Illinois wastewater surveillance system, the counties across the state of Illinois. And then in Chicago, where we're at 7 wastewater reclamation plants, 6 sites that serve facilities like I mentioned, Cook County Jail, O'Hare Airport, and then a number of sub-sewersheds. So I'm talking like in the sewer system. We sample in manholes in the city. So we're about 18 sampling points total. This changes regularly, but this is just about where we are now. We are, like I mentioned, we submit our data to the CDC. They recently, in the last couple months, they started, a couple months ago, they started incorporating wastewater data into their COVID data tracker. And this shows that all these dots in the state of Illinois are ours, which is very exciting. You can see that there's a strength in the Midwest in general for wastewater surveillance, and then at least in terms of reporting to CDC. But we like to kind of look at this and pat ourselves on the back that we're pretty well represented on the COVID data tracker. So our sample collection, we obtain samples. Most sites we get twice a week, and when we get them from water reclamation plants, most of them are 24-hour composite samples. That's what's shown in the upper left picture, an auto sampler and the sample being taken there. On the right is the box that the plants around the state use. They ship the samples to my lab. I'll explain the method transition and the transition to my lab in a moment, but they send the samples to my lab by FedEx overnight, and then local Chicago samples from both MWRD and sewers, jail, airport are collected throughout the week and just delivered directly to my lab. We work closely with both current and with a contractor who helps sample. We're transitioning from, this is, I don't know if you can tell, it's a tiny picture, but they're pumping water from the sewer water from the manhole. We've developed some rigs that we have deployed throughout the city that serve as passive samplers that we just leave in the sewer and then retrieve. So the methods have, like everything in the pandemic, right? Our methods have evolved since we started in 2020. This is Abhilasha, by the way, from the School of Public Health. So everything initially took place in her lab. This picture is of Christopher Owen, a PhD student in my lab, who worked in Abhilasha's lab for the first year of the pandemic. And so samples were sent directly to Abhilasha's lab and extracted there. You can maybe see there's a filtration rig in the hood here. So we could process maybe 8 samples at a time. We filtered the wastewater, and extracted, not DNA, we extracted RNA, and did qPCR in Abhilasha's lab. And this method worked remarkably well. There was a lot of ramping it up and developing it, and troubleshooting it, but we had a pretty good system going, but we were kind of tapped out because of the nature of the method, which, you know, filtering takes a long time. It could take an hour or more to filter 50 milliliters of wastewater, depending on the components that day. And the rig could take maybe 6 or 8 samples at a time. It took a while. And so as we talked about expanding the program, we knew that we couldn't expand it using these same methods. And so as we moved into the statewide program, we scaled and incorporated a good bit of automation. So these are some of the key players who are involved in my lab currently. The first 3 are all folks who we hired to maintain this program. Adam is our lab manager. Chiyu and Jarju are both technicians in the lab. And then we have some graduate students who are involved. And again, this is a small sampling of the people who are involved. It's a large effort with many players, but this method has allowed us to automate a lot of the steps and also to scale the process so that we can extract from 24 samples at a time, and we can extract from 24 samples in 30 minutes, whereas before we were doing 6 in a couple of hours. So it's made a huge difference. The new method involves these plates where we transfer wastewater. We extract from 10 milliliters of wastewater. We put them in these plates with these magnetic beads. And so the first step is a concentration step. We use these nanoparticles that are magnetic that pull out viral particles from the wastewater matrix. And then we extract RNA also with a magnetic bead-based approach in 96 well plates. So we can extract 96 samples at a time, again, in under an hour. And we, instead of qPCR, we do digital PCR in my lab. The difference is not crucial for the purposes to convey today, but in general, it's more sensitive, and this is one of our instruments that has can take 4 plates at a time. So again, it helps with the sensitivity, but also with the throughput. Interestingly, the CDC has recently announced that they're trying to work with NIST, the National Institute of Standards and Technology, to formalize or incorporate some standards into wastewater epidemiology in general. And as part of that, they're pushing everybody to move to digital PCR. And we have a number of targets. We look for various components, N1 and N2 are regions of the SARS-CoV-2 genome. BCOV is a bovine coronavirus that we spike in and use as a recovery matrix. PMMOV is a pepper virus, actually, that turns out to be a really useful fecal indicator. It was actually first used as such maybe 10 years ago or so in urban streams to identify fecal loads. And because it's so widespread and it's such a big part of human diet that there's a lot of it. And it ends up being a good marker. We've also built in assays to target various variants, that's funny to say, variants of concern and variants of interest. And we keep building on this. I'll sort of end with additional targets as we build this program and expand. So this is some older data from 2020 and 2021 that just kind of shows correlations between case data and wastewater data. So on the left is epi data, cases, case positivity rate in gray, and then tests, number of tests in blue. And on the right, we have various gene targets. So N1 and N2 are both indicators of SARS-CoV-2, and we see similar trends. If we overlay the two with each other, we see a pretty good relationship between the prevalence estimate based on case data and the quantification that we get in wastewater. We've also, actually, CDPH put together a report and they included these comparisons between wastewater data and case data for a number of sewer sites around the city. And so also showing that there's a good, that the wastewater data tends to reflect case data. And this is also pre-Omicron and also masks coming off. So that's another thing that I didn't mention at the start, but there's, as many people probably know, a lot less testing is happening, and fewer test results are reported to public health agencies. So there's people are relying more and more on home rapid antigen tests, and those that don't get reported to any public health agencies. So now that there's more of a disconnect between case data and actual data, and the beauty of wastewater surveillance is that that doesn't matter. So while the case data may become less reliable as or the test data may become less reliable as testing frequency goes down, the wastewater data, the reliability of wastewater data is maintained. So what we do is we report trends, and they look something like this. So we report 2-week and 4-week trends, and we report this to IDPH and to CDPH, and we'll say, you know, some site may have an almost certainly increase or a very likely increase, or we're not certain of the change because we don't have enough data points, or the trend is not apparent, or it almost certainly looks like a decrease. So we have various thresholds that we define the trends. And this is based partly on our own data analysis and partly on CDC recommendations of looking at trends in wastewater data. I'll also point out that Christopher, PhD student in my lab, just published a paper a couple weeks ago where we looked at comparison between influent and effluent. So what comes out of water reclamation facilities in Chicago. And part of this was came from previous interests of mine pre-COVID, looking at how effluent helps shape microbial communities in receiving bodies of water. But also just our curiosity in whether there's RNA from this virus in that makes it through the wastewater treatment process. And I won't spend a lot of time on this, but the take-home message is that the red is the effluent, and so there's most of the RNA, SARS-CoV-2 RNA, is removed through the treatment process, but we still sometimes do detect SARS-CoV-2 RNA in effluent. That's not to say that it's infectious or it's whole virus or any of that, but it does make its way. Some of it can make its way out of the water treatment process, and so, you know, from my microbial ecology background and interest, we think a lot about, even if it's just RNA, how it might shape receiving communities, microbial communities. We also, what was included in this paper and what's on these plots compares whether there's a tertiary treatment or a final disinfection or not. So yeah, happy to talk about that another time in more detail. I did want to mention our sequence data because I think this is a real strength of our project, and a lot of all the respect goes to Sarah Owens at Argonne National Lab, because at the beginning of this there was a lot of doubt whether we would be able to sequence this virus out of wastewater, and Sarah also has a background in looking at microbial communities in complex environments, and George and I and others have worked on on microbial and viral communities in wastewater before, and had a feeling that we would be able to do it, but there was a lot of doubt in the public health world whether we would be able to get any meaningful sequence data out of wastewater. And it turns out to be probably one of the most valuable aspects of our work. So the plot on the left shows the light blue is Delta and the orange is Omicron. So we could see the arrival, and it's roughly from left to right, it's roughly time. So we could see the arrival of Omicron in our wastewater data in early December. The plot on the right shows a breakdown of sublineages and other variants. Right now we're up to over 100 sublineages of Omicron, and we can detect a lot of them in wastewater, and some of them before they're seen in any clinical remnant sequence data sets here in Chicago and across the state. So I think even now, I think this data has become very valuable. We work kind of closely with the Ripple Lab, which is at Rush University. They're doing clinical remnant sequencing in of Chicago samples, and they're the heavy black line on this plot is their data. So they're showing the proportion of Omicron from late November to beginning of January. And plotted with that is our wastewater data. And we can see that there's some sites where we can start seeing, where we started seeing Omicron before case data, before we started seeing it in clinical swabs. Another thing to keep in mind is that with, especially in the early days of Omicron, a lot of the clinical sequencing was happening on very targeted samples. So there was, they'd get a lead that somebody, there was a suspected case, and so they would sequence from a specific person. Again, we don't change our sampling routine for wastewater. We're always getting everybody, essentially. Not quite everybody, but we're always getting this a lot of people. And then, you know, some of our bigger plants like Stickney, it's the signal lags, and I think that's just a complexity of a sample that from a facility that serves well over 1 million people. So I'll finish up by talking about what our public health agencies do with the data. They use it in collection with other epidemiological data. You know, we report actually multiple times a week, but we brief our public health agencies, IDPH and CDPH weekly, and they use this data with other data to help improve local decision making. And so I think it's important to note that wastewater data on its own is not being used to make big decisions, but it's used to sort of validate other data that is collected, and to make up for missing data, like when we have fewer reports of testing. And so we cover gaps in clinical sequence coverage. And again, the last point, helping to track variants, I think is a really important part of this work. So we continue to, believe it or not, we continue to grow and develop to apply our regional mapping with uncertainty models, trying to understand the sensitivity and the uncertainty around some of our values at different scales, which I'll mention in a second, and also to calibrate and integrate models with regional data, and to take all of this at different scales to better understand how our data can be more meaningful, or how it can be applied. And so the question of scales is really illustrated well with our program, because in across the state, we're looking at the largest population centers in each county, or most counties. We had anticipated we'd be in every county, but turns out not every county across our state even has wastewater treatment plants. And then in Chicago, we have the Chicagoland area where we have the major wastewater reclamation plants and this broad coverage. And then in the Chicago, in the city, we have sub-sewershed sites. And so the very last thing I wanted to talk about was that I kicked off with this idea that we're thinking beyond COVID-19, and I actually took COVID-19 out of the title. And so on the right is a plot from work that came out of my lab pre-COVID. We've been for a while interested in things like antibiotic resistance genes in urban urban freshwater systems. So we've done work in Lake Michigan and in the Chicago River, looked at response or the effects of storms on antibiotic resistance genes. In Tempe, Arizona, even pre-COVID, there was work being done to monitor opioids in wastewater. And we have partners at the Chicago Department of Public Health who are really interested in this. We're actually involved in a pilot study. We're sending, MWRD is sending samples to the University of Florida for an opioid surveillance pilot. And we were just talking about this in a call I had right before this about this monitoring in Israel of a silent polio outbreak. That this was also pre-COVID. So, we knew that wastewater epidemiology could be useful for monitoring of other public health concerns. But I think after COVID, it's really come to the front as being a really good way to understand community health. We can get rapid, sensitive analysis of entire communities without conveying personally identifiable information. And this was the focus of the meeting I had right before this. We're talking about ethics of wastewater surveillance. So CDC has presented their thoughts on the future of wastewater-based epidemiology and future potential targets, things that are shed in stool or would show up in wastewater, that are nucleic acid based and stable enough that we can detect it in wastewater. And so they have building a list of potential targets, and then things like antibiotic resistance genes that are also nucleic acid based, but not pathogen specific, but definitely, WHO and a major public health concern of antimicrobial resistance. So we are working to once we have this infrastructure in place now, and we're working on future targets and the next pandemic, monkeypox, right? We actually have a monkeypox assay on its way to our lab to see if it's even possible to detect it in wastewater. But yeah, the idea that we can use wastewater to monitor urban and wastewater systems for lots of public health concerns, both pathogens and things that are not pathogens, but other public health concerns. And ultimately what kind of fits in with the theme of these third coast water talks is, focusing on actions and response, things that we can do to keep our nearshore water quality safe and sustainable and keep our communities healthy. And with that, I will be happy to take questions.
Thank you so much, Rachel. What an amazing program. It's really nice to see it come to such fruition after a couple of years, and seems like Chicago really kind of led a lot of the early efforts in early 2020 with some of that seed funding. We have a couple of questions that have come in. Excuse me. A couple you have answered, thankfully. There was a question about applications beyond COVID, right? And you and I actually had a nice conversation at the IWPC conference recently, where you presented this work, and you were expressing your enthusiasm for, I guess, being able to share and collaborate in a similar way on other topics that, COVID has allowed us to frame everyone's attention and feel comfortable and confident speaking and sharing data before it's been completely vetted. And you're expressing how wonderful it would be to have that kind of collaborative nature in other areas, and what could we do with that? So that was a fun
Right.
conversation.
You know, I hope it lasts too. Like I feel like a little bit of it is waning now that everybody's got established, or a lot of places have established programs. It's not quite as active as it was in the beginning, but we were all in such a frantic place at the beginning of the pandemic, trying to get things in place, especially because we kept thinking about how, if this infrastructure was in place before COVID happened, our response could have been different and maybe better. And, you know, there was a lot of struggle early on to find out when was the first case in my community? When was the first case in the United States? How did it move? And now we could do that with all this infrastructure in place, but it wasn't there to begin with.
Yeah, excellent. We have a question just about, in general, future collaboration. Do you offer an email address, or is there a generic email address for this program where folks can reach out and ask more questions about the program or get involved?
Sure. You are welcome to contact me, but I will also put, I have to get the address here. I will put it in the chat. So Laura Clements is our program manager, and everybody is involved in this project is amazing, but she's really a good person. She and Crystal, hold on. And Crystal are both good people to reach out to. If there's, we're bringing in, new counties all the time. Each week we add new facilities. So Laura is the first one, Crystal is the second one. We keep bringing in new counties. We're talking about whether this is something that can be done at other congregate living facilities. Yeah, if there's interest in your community, or if you know of somebody who's interested, or you want to get involved in this work, or you think you have something to contribute, by all means, reach out, and here's me.
Thank you. A couple of questions regarding wastewater treatment. You had shown a graph of influent versus effluent in the RNA that's left behind. Do you have any idea or any thoughts on what's happening with that RNA? Is it being removed due to adsorption, to biosolids? Have you looked at any of the biosolids in the area?
No, we've talked about this. I've talked about it with folks at MWRG. I've talked about it with George Wells here. Yeah, it would be really interesting. We've done some work in the past on looking at various stages in the treatment process for things like, so antibiotic resistance genes also get attenuated through the treatment process. So we see some that do get discharged, but the amount gets reduced a good bit through the treatment process, but we've been able to look at various stages along the treatment process, and it's always been a little bit outside of the scope of this current work. We've only looked at influent and effluent, so here I could only guess at what stage it's being removed, but it's also RNA, so it's in theory it's less stable than DNA anyway, so some of it might just be destroyed because it's RNA, and some may be removed through the treatment process, but I don't have a good answer, and I also would like to know.
Well, there's a number of studies going on with biosolids, so I'm sure there are some good resources out there we can point folks to. Another question regarding specifically Stickney. Do you have any thoughts on why there's a lag in detection at that plant? Yeah, so Stickney is our largest water reclamation facility, the largest in the country, right? And I think it's just a signal to noise issue. There's so much there that I think our ability to detect so in the beginning when Omicron first showed up in Chicago, it was only a few cases, and I think to be able to detect just a few cases in such a complex sample is really hard. So it's the smaller treatment plants and the sewers where we saw it first. But once it gets over a certain threshold, we see it, and we see it really well. But I think, most of what we sequenced from Stickney at the beginning was
Delta because that's what was more abundant. I think it's just signal to noise.
That makes sense. A couple of more public health related questions. Do you have a sense for how the public might be using the data out of this program? There was a comment made early in the program about the difficulty of explaining some of this to the public. And so do you have any thoughts on how the data might be used by the public that's being captured throughout Illinois right now and presented by CDC nationally?
Yes, that's a really tough one to address. You know, different states and different, I'll just use the US as an example, different states and different cities across the US have had a really different approach to disseminating their wastewater results to the public. And part of that is really related to constraints from the local municipalities, support from local government, and state government. And there's some cities where as the data is generated, all of it gets posted on a website for anybody to see with detailed FAQ and an explanation of the data. And in some cities, it's not shared at all. And I feel like here we're really working hard on how to approach that, how to disseminate the information. We don't have a big public facing dashboard with all the information released, but it is available now through the CDC. I think the CDC data can be a little bit hard to interpret. So a good example is at the beginning of not even in the beginning of this wave, but when in that brief quiet time between the first Omicron wave and the current one, where cases were really low and all the masks came off, the wastewater data across the country, those dots on the CDC map started turning red. And there was a lot of public response that was like, why are the dots red? It's going up, it's going up. And we had to convey that in a lot of cases, it was going up from nothing to just a little bit more than nothing. So when we say like, it could be 100% increase, but it doesn't mean that there's a lot present. And so I think how to interpret those data and how to convey that is a challenge. And we think about that a lot and wonder about how best to communicate it and how it can be effective and useful without being misleading people or making, I don't know, making people worried prematurely or any of that. So I think that didn't really answer your question, but the big answer is that we're working on how to convey that, and we think about it a lot.
I think public health communication related to scientific data of any kind is incredibly complicated and very, very, very difficult to navigate. One last question surrounding detection limits of infection in communities. You know, what could a detection limit be in a community before you start seeing, you know, something at the plant? And then conversely, are we any closer, or not conversely, to follow on that, are we any closer to using this data to pinpoint infection rates in a smaller place or even at household? It sounds a little tough there, but
yeah. So household is tough. I think it's possible, but that starts tiptoeing into a lot of ethical critical areas where we start, you know, a lot of what I presented here today was the foundation of the wastewater approach is that it's generally anonymous. And once you get to a certain level, it becomes less anonymous. So as an example, CDC will only post dots from counties that are above, I want to say 10,000. I forget, there's a population, somebody on this call knows better than I do. There's a population cutoff for when they'll start showing dots, because when it gets to a too small community, 3,000 people. Is it really that few? Well, yes, okay. So when it gets to a population too small, they're worried that you can start getting to levels that it gets less anonymous. Yeah, so I think it's possible, but it's not really practical to get that level. Although there's lots of talks of you know, smart toilets that can start monitoring things about, but that needs buy-in from households. That's not something that we can start knocking on doors and collecting samples.
Okay, thank you. And you know what, I'm going to squeak one last question because it came in, and we are here to talk about innovation and bringing our sector forward with technology, and this is super related. You know, could you comment on the potential, and I'm going to just read it, for automated pathogen detection in situ in sewers or wastewater treatment plants? Do you see a future perhaps where pathogen quantification or even sequencing occurs directly at the plants themselves rather than in these sophisticated labs, maybe a lab on a chip or something you can do on site?
Absolutely. I think there's, you know, initially we had involved STEM Loop in our initial research proposals, a company developing lab on a chip technology. And there's other commercial ventures out there that are trying to do this. It has to, I mean, it has to be automated enough. I didn't mention this in the talk, but all of our wastewater facility partners are voluntarily doing this for us. And we can't, I don't feel like we could ask too much of them. Some places there's like one guy who maintains the water reclamation facility and is close to retirement, and he's doing this on a Saturday. He comes in to get a sample for us. And so I think it's a great question, and I think things have to get to a point where we can, you know, stick a contraption in, and it's got to have a way to report the data and be as hands-off and reliable as possible. But I think there's a lot of development and innovation happening in this realm, for sure.
Thank you for that. And we're going to go ahead and wrap up. I'm going to go ahead and share my final screen. I think.